Okay, so most of the guys are telling it says answer A. Okay, so yes, the correct answer is option A, which is basically iron. And we all know that iron is present in our uh, uh, in microorganisms, uh, and therefore the iron has a pretty much role in our body also, in presence of hemoglobin. And so basically what happens in the process is like the transport of iron is done by transferrin, and then that transferrin transfers it to various other molecules in our body which need iron, let's say hemoglobin or let's say myoglobin, depending on the quantity of the need. And then there is one more protein known as ferritin, that will further store it, and then that storage can be further used if we have a iron deficiency in our body at a later stage. Yeah. Okay, so the next question is about okay, so in enzymes in which metal act as active center are called as what? So we all know that some enzymes do have a metal center, some enzymes do not have a metal center. Uh, but the thing is, what do we call those enzymes which do have a metal center? So you guys can guess the answer, then we can discuss about it. So obviously the enzymes in which metal act as active center are known as metalloenzymes. So as it is pretty much obvious, so the one of the very good example about metalloenzyme is known as nitrogenase. So nitrogenase is a metalloenzyme which catalyzes the breakdown of obviously nitrogen molecule to form ammonia, and we know that nitrogen to ammonia is a pretty much good reaction in both industrially and also mm, by biologically in the case of nitrogen fixation. And so the catalytic site of this nitrogenase enzyme has 7 iron ions, and if you guys see the structure looks like something like this, where we have this disulfide iron moiety as one of the core moiety, and there is also other moiety which has this FeS4 cluster, and there is one more disulfide moiety which has 2 iron, and then you also have a molybdenum in the inside the active site. And therefore this is basically an equivalent to the Haber's-Bosch process that people use industrially for the formation of ammonia, and we know that ammonia is pretty much required industrially for various purposes. And therefore this nitrogenase understanding this nitrogenase enzyme is really important for us. So nitrogenase is a metalloenzyme and it has two metals, one is iron and other one is molybdenum. Okay, so the next question is about ferritin. So ferritin, what is the role of ferritin? As I have previously also discussed in the first question, what is the role of ferritin? So as we had previously discussed that there are two enzymes. One is known as transferrin and the other one is known as ferritin. So transferrin transfers iron and ferritin basically stores iron. And so therefore the correct option is option B. So if you see our basically our uh, RBC, our red blood cell, it contains heme, and heme stores iron in iron 2 oxidation state, and then all the and therefore what happens is, uh, the ferritin, which basically stores iron, it can give the excess iron to various enzymes like lysozyme, and then further those enzymes will further give that iron to various other enzymes, let's say to transferrin, to ferroprotein, and therefore the iron storage can be used further in the later stage whenever it is necessary to be used. So therefore the role of ferritin is to store iron, and the role of transferrin is to transfer iron, and obviously heme is the very basic moiety which stores iron as a iron 2, and we know that heme moiety is really essential for us because it is the moiety which uptakes oxygen from the air that we breathe and transfer it to the um, red blood cells. So the function of so what is the function of ferritin? So the function of ferritin, the first option is to carry oxygen, second is to regulate secretion of iron, or to regulate secretion of zinc, or none of the above. So the function of ferritin is obviously very obvious from the previous slide. It is to regulate secretion of iron. So if you see ferritin, how does the ferritin moiety is constituted? So it is basically so how does it basically regulates the whole process? So if you see the iron 2 plus that is coming from transferrin, which is basically giving us that, which is basically transferring the iron 2 plus, it get inserted in the active side or the catalytic side of the ferritin molecule, and that is this molecule, and then uh, it is getting oxidized in the iron 3 plus stage and basically stored in the iron 3 plus stage in ferritin, and whenever it is necessary it will further remove it as iron 3 plus only, and therefore this is one of the example of ferritin that has been found in frogs, and the basic structure in ferritin is basically it exists as a dimeric form where you get two iron molecules, one in iron 3 oxidation state, and here you can see various amino acids molecule that is basically bounding the active side of the ferritin molecule. So if anybody has any doubts so far, in any of the questions or any queries, they can ask or type in the chat box and we can discuss. Otherwise, we will move ahead to the other few questions. Yeah, somebody has any question, they can ask. Yeah, what is the question? Yeah, yeah, yeah. Yeah, obviously, it is an independent process. It stores iron in iron 3, and then ferritin can basically regulate depending on the need of various other enzymes that is present in our body that requires iron in various oxidation states. So let's say, yeah, let's say after some time there is iron deficiency in our body due to various vitamin deficiency or something, then at that stage our body may have stored iron in ferritin, and then it can basically transfer to various other molecules like hemoglobin and myoglobin, and so basically uh, that is the moiety that stores iron in iron 3 state. I hope I'm clear. Okay, so let's move on to the other few questions. So the next question is, in transferring the oxidation state of iron is, obviously I think I've already discussed about it. So if you see the active side of transferrin, so transferrin has a various amino acids in the active side, and it contains iron in iron 3 state. So in the active site there are two groups, that is tosyl group, which has a phenolic OH bond, and there is also a histidine molecule and further a aspartyl molecule. And in the active site the iron basically binds in iron 3 stage, and there is also a carbonate molecule in the active site which further enhances uh, the binding of the iron in the active site uh, by your CO bond, and further the iron you can see here is a hexa coordinated iron. And so the iron in transferrin exists as iron 3 state, and basically this is the molecule which transfers iron to the various uh, other enzymes in our body. So the next question is about the iron carrier in microorganisms is obvious is what in microorganisms. So the iron carrier in microorganisms is known as siderophores. So the so if you see, transferrin is basically present in few organisms, not in microorganisms. So it is present in mammalians, and the in microorganisms basically siderophore is the very basic moiety which uh, basically carries or you can say transfer iron from inside microorganisms. And if you see the basic structure, the structure is basically a hexa coordinated molecule. Only here also you have let's say uh, various OH phenolic OH moieties, and there is a citric acid moiety which binds the iron, and here also the iron uh, is in hexa coordinated stage, and there is also one paper that I have cited, you guys can see for it. Okay, so the next question is about carboxypeptidase. So the active metal center in carboxypeptidase is what? So if you guys know about carboxypeptidase, what is its role, and definitely what's the active metal center that is present in carboxypeptidase? I think some of you guys can guess from the name itself. So if you see carboxypeptidase, so the correct option is zinc. So in carboxypeptidase, the active site contains two histidine molecules, and it also has a glutamate molecule which binds zinc in the active site. So here the zinc is being basically there is also a water molecule that binds with the zinc, which has a very crucial role in the mechanism of the reaction. So carboxypeptidase have exist in two forms. One is carboxypeptidase A and one is carboxypeptidase B. And carboxypeptidase A basically cleaves various peptide bonds. And if you guys know about peptide bonds are bonds with various amino acids, long chain amino acids, and it has a C terminal and it also has a N terminal. And so it basically cleaves peptide bonds or the amide linkage in the peptide bonds. So it contains zinc iron for doing this mechanism, and it contains zinc in plus 2 oxidation state. Okay, so we can move to the next question. So the metal ion responsible for nitrogen fixation by the enzyme nitrogenase are. The first option is cobalt and iron, copper and iron, your tungsten and copper, and iron and molybdenum. So obviously the metal ion that is responsible for nitrogen fixation, as I previously also discussed, is iron and molybdenum. So here what happens is it exists as a iron sulfur cluster where there is a iron that exists in so I can show you so in this structure if you see so if you see the FeS4 cluster, so there is Fe4S4 cluster. So you can see that in one of the cage there is 4 iron, that is 3 iron in iron 3 plus and 1 iron in iron 2 plus. This is one of the cage, and there the other cage is also connected to the other cage by your sulfur linkages. And here there is also one molybdenum moiety that is present in this further binding of these two cages. And so therefore there are a present of two metals, iron and molybdenum. So you have both iron in plus 2 and plus 3, and there is also presence of molybdenum which is present in plus 3 oxidation state. So this is the whole overall structure of nitrogenase enzyme which contains two metals, iron and molybdenum. And as I have previously also told that nitrogenase is basically the enzyme fix nitrogen to ammonia, and the alternative of this biological enzyme is our Haber's-Bosch process, where industrially we form ammonia from nitrogen. But in that process, as you guys, if some of you guys know, in that process we need very high temperature and very huge amount of pressure to form ammonia, because breaking of the nitrogen molecule which has a triple bond is very hard. And but if you see enzymes which do it very with huge turnover numbers and very efficiently. Okay, so we can move to the next question. So, as I previously also told about this siderophore, so it has iron in plus 3 complex. So, if you guys have to, so the overall structure of this siderophores is what, and what is the spin configuration of the iron 3 plus in siderophore. So, if I have previously also shown you guys the structure of say siderophore, so there here the iron is also present in hexacoordinated species, and it is a plus 3 species. So if you guys know about crystal field theory and splitting, how the d electrons are splitted, you guys can guess out about the spin state and the geometry of the iron in siderophores. So I will give you a couple of minutes to decide. So if you see siderophores basically is a hexacoordinated. It has iron. So if you see the active side, this is the active side. I have shown you the crystal structure of the siderophore, and here the iron is linked by various oxygen moieties that is present from various amino acids, and here it is basically octahedral molecule, which is as obvious, but it also contains, because it is iron 3, so it will be a D5 configuration, and whether it is a high spin or low spin, that you have to figure out from the crystal field theory. So if you see that it has oxygen molecules, so the iron 3 plus, that is your D5, and we know that oxygen is a weak field ligand as compared to nitrogen, and if you see the spectrochemical series, so therefore it will be a high spin D5 configuration in spite of a low spin D5 configuration. So here the iron is in iron 3 D5 octahedral geometry with a high spin or high magnetic moment of the iron 3 plus. I hope so. Let's move to the next question. The function of carboxypeptidase A is to what? It carry oxygen, store zinc, store iron, or to hydrolyze a peptide bond at the carboxy terminal or the C terminal end of the peptide bond. Okay, so we all know that carboxypeptidase is basically used to hydrolyze peptide bond, as I previously discussed. So it basically cleaves the bond on the C terminal of the peptide bond. So if you see this moiety here, um, so here what happens is, this is the C terminal of the peptide bond. So let's say this is the last amide bond in the whole polymeric chain, and this is the C terminal because the carbonyl is inside the is towards the inside of the chain, and the NH of the amide bond is outside. So therefore it is the C terminal of the amino acid side chain, and finally we get a n minus 1 polypeptide, and one of the amino acid side chain has been cleaved. So carboxypeptidase A, the role of it is to basically cleave peptide bond from the C terminal end. And obviously we also know that it contains zinc in plus 2 oxidation state, but the basic role of it is not to store zinc, but the basic role is to hydrolyze peptide bond. The storage of iron in human body is carried by what? It is about human body. So if you see, the storage of iron in human body is also done by ferritin only. So ferritin is also present in our body, and we all know that myoglobin and hemoglobin has its own role. Hemoglobin is for hemoglobin and myoglobin both are oxygen binding proteins, and cytochrome P450 is basically not present in our body. It is being present in various other organisms, And here in ferritin it exists in the iron is basically in the uh, is basically used to store in our body, and you can see that there are various glutamate, histidine, moieties which basically bind the iron to store to stabilize it. Okay, so the next question is myoglobin is uh, so let's move to myoglobin and hemoglobin and understand it in a much more better way. And so if you see myoglobin, so myoglobin is a catalyst for epoxidation reaction or component in photosynthetic system or a nitrogen fixation enzyme, or it is a dioxygen binding metalloprotein. So I think most of you guys know about it. So myoglobin is a dioxygen binding metalloprotein. So if you see the overall structure of myoglobin or maybe even hemoglobin. So hemoglobin is just a tetramer. It exists. It has three porphyrin systems. So it has three irons. And myoglobin it is a monomer. So the overall structure is that there is a porphyrin molecule which has this nitrogen atoms which can strongly bind to iron in the in a hexacoordinated way, and there is a other histidine molecule from the side chain of the overall protein which can basically bind from one of the lower phase to stabilize the iron, and then it can basically bind with the oxygen molecule uh, that we are inhaling from the air, and it will be hexacoordinated species. So this is a very basic general structure of myoglobin here. So it is a dioxygen binding metalloprotein. Okay, so I think it's clear. So let's move to the next question. So which of the following statement for hemoglobin is incorrect? So if you guys know about hemoglobin, so the first is the binding with oxygen is weaker as compared to myoglobin. Iron is 5 coordinated. Yeah, please mute if. So here the iron is 5 coordinated, or iron is coplanar with the porphyrin ring in the absence of oxygen, or the other option is oxidation state of iron is +2. So which of the following is the incorrect statement, not the correct statement? So if you see, the correct answer is actually option C. So most of you guys maybe have thought about option A. So so if you see how the iron is basically there in the hemoglobin is so it basically binds with oxygen. So when there is no oxygen present, uh, like, and at that stage or the deoxygenated state, the iron is not in the plane of the porphyrin ring. It is basically towards the histidine moiety, which binds strongly towards and pulls it outside the plane. But whenever whenever there is a presence of oxygen, the iron has been pulled towards the plane of the heme, and therefore iron is basically not coplanar when there is absence of oxygen. So the option C is the incorrect one. Obviously, iron is 5 coordinated, and oxidation state of iron is +2. And obviously, if you see about the comparison of oxygen binding of myoglobin versus hemoglobin, obviously in hemoglobin there is like 4 oxygen molecules that it binds, but that is not saying you that it binds more strongly as compared to myoglobin. It is actually the opposite. If you see the binding curves, so in myoglobin the oxygen saturation is done at a very huge scale as compared to hemoglobin. And in hemoglobin there is a cooperative effect which comes after a certain uh, pressure of oxygen that has been present. So actually it is the opposite. The binding constant, if you see, of hemoglobin versus myoglobin. Obviously hemoglobin takes 4 oxygen molecules because of the cooperative effect at a later stage. So the incorrect option is iron is coplanar with the porphyrin ring in the absence of oxygen. Okay, so let's move to the next question. Hemoglobin is an oxygen carrying protein. The correct statement about oxyhemoglobin is what? So this question is about the oxidation state of iron in oxyhemoglobin, and also the oxidation state of the oxygen that has been bound. So if you see in hemoglobin, the oxygen carrying protein, the iron is basically in the. So if you see the deoxyhemoglobin, it is in iron 2 state, but when oxygen binds with it, it comes to iron 3 state, and it a- and at that stage it basically comes within the plane of the porphyrin ring, and so therefore the iron is in plus 3 oxidation state, while the oxygen molecule which is bound to the iron, it becomes peroxy molecule and it becomes O2 minus. Therefore, the correct option is option A. And also there is one more histidine that is much more distal position, that is much more further from this porphyrin system, that also bounds to the oxygen and helps it to go to the O2 minus form. So I think it's clear. So let's move to the next state. So it is about the methemoglobin. So what is the oxidation state of iron in methemoglobin, and why is methemoglobin important and other stuffs?